Welcome, everybody. You've reached the NYU session today on expanded access and xenotransplant. I'm Art Caplan. I'm the director and head of the Division of Medical Ethics here at NYU Grossman School of Medicine. And I, along with colleagues, including Tamar, Schiff, who you'll meet in a minute, have been running a project on transplant ethics for a number of years here. We've had the opportunity to work with some of our colleagues in the surgery department here in Transplant Center who are interested in xenografting. I think many of you on the call are well aware that there is a big shortage of organs. And obviously one strategy for trying to solve that shortage is to look toward animals. People have been looking toward animals actually for decades to try and solve the shortage of organs. I've been around so long that I was actually involved when a baboon heart was transplanted to an infant at Loma Linda University almost 40 years ago in an effort to see if they could bridge that infant who was dying to a human heart transplant, and it did not work because of rejection, immunosuppression problems. Some of that has been attended to by attempts at genetic engineering. We find ourselves moving toward pigs. As one of my students once said, a pig is kind of a human being horizontal, right? And they meant that in terms of physiology, size, and comparability of organs. If you're in the primates, not only do you have some pretty severe ethics objections to their use, but you also have size issues. They're just not baboons, monkeys. These animals might be useful in very small infants, but they're not going to support 200-pound adults and so on. Pigs are also animals that can be bred quickly in pretty big numbers. The animals that are bred for this type of strategy are raised in very clean and humane conditions because they don't want them sick and they don't want them stressed. Doesn't mean there's not an argument to be had about still should they be sacrificed to try and save human lives. But animal suffering, I would say, in this area, as opposed to the factory farming of pigs, is much more humane. Genetic engineering is a little bit of an experiment in itself. People have ideas about what particular mutations might change the immunogenicity of a pig's kidney or liver to make it easier for a human to accept, but it's still not certain. There are obviously issues that surround viral transmission from pigs to humans, carrying them along and maybe out to others. But there's also an interesting challenge about how you even make progress from testing these experiments in animals into getting to the point where we're ready to trial them in humans. And that's our focus for today. What is the best pathway? Do we need a different pathway? Or existing pathways acceptable in terms of developing research that might lead us to a viable xenograft of a pig organ into a human being with the hope of clinically significant survival? I'm laughing a little bit when I say that because that's also controversial as to what would be a outcome that would be not only recognized as significant by recipients, but also by payers. So that'll be a topic perhaps for another day. But we have some really great folks assembled to talk to a little bit about pathways that can be used in trying to develop research in this space. You'll hear in a minute about expanded access, which is one route to go, and somewhat kicked around in terms of different definitions. And you'll hear other comments later about perhaps developing a slightly different path to go down. These paths always involve issues around transparency, informed consent about risk and benefit, making sure that people understand what it is that's the research behind the attempt to try this when they agree to be a first in human or a third in human recipient, what preceded this, why is it believed to be safe, who reviews it, IRBs, special committees, nobody except the surgery center. And we're also alert on the call, although maybe reserved for Q&A, to the fact that there are other nations besides the US who are interested in xenografting and have their own ideas about how to approach and handle experiments in humans, and then shifting over more to clinical trials, and then ultimately to efforts at therapy. So without further ado, I'm going to turn to my colleagues here at NYU, Jayne Kim and Karen Khalil, and I'm going to have them just, Khalil, excuse me, and have them just introduce themselves briefly to you. And each of these speakers will go, they're going to share a little session of about 5 minutes each, and then we'll move on to Dr. Kumar of the FDA, and then another one of my colleagues here at NYU, Tamar, Schiff. So let's start with, I think it's Dr. Khalil.
Thank you, Dr. Caplan, and thank you for the introduction and for the opportunity to be here. My name is Karen Khalil. I'm the Xenotransplant Project Manager here at NYU, working with a large team to oversee all the work that we've been doing so far in xenotransplantation, which we'll share with you today. I'm a clinical transplant pharmacist by training. And Dr. Kim, do you want to introduce yourself as well?
Sounds good. Thank you, Dr. Khalil. Hi, everyone. My name is Jayne Kim. I'm the Assistant Director of Regulatory Affairs here at NYU. I lead all FDA initiatives along with my team. So we do FDA submissions, any sort of commercialization efforts within regulatory affairs.
Thank you, Dr. Kim. And I think we'll start with some very brief background. I think Dr. Caplan basically covered this, but I think we all are aware that there's an unmet need here in transplantation, and that there are over 100,000 people on the wait list, and thousands die each year waiting for a life-saving organ transplant. And there are numerous different avenues that are currently being explored for creating a renewable and sustainable source for human organ transplantation, including artificial organs, decellularization and recellularization of pig organs, which includes kind of a scaffold of the organ that is recellularized with human cells. And of course, xenotransplantation, or utilizing these pig organs for human transplantation. So again, I think Dr. Caplan touched on almost all of these, I believe. And but why are we using pigs? Of course, pigs and humans have been living in close proximity for years. They have great breeding potential, widely available. Of course, this varies depending on the types of genetic modifications that you do in these pigs and that you make, and that affects the viability. Their anatomically, kidneys, for example, are similar in size and structure. And actually, given that pigs are more genetically distant from humans compared to primates, for example, porcine viruses might not have the necessary genetic adaptability to infect humans as easily, so lower risk for disease transmission. And of course, as Dr. Caplan mentioned, there's a lot of experience using CRISPR technology here to genetically modify these pig organs to make them more compatible with the human immune system, as well as to reduce infection risk. And from an ethical consideration, of course, over 100 million pigs are bred and consumed annually in this country alone. So there is a significant amount of research that's been done in xenotransplantation in the non-human primate model. However, there are several limitations leaving us with critical unanswered questions that we need to elucidate prior to moving towards clinical xenotransplantation in multiple living patients. And these include what immunosuppression medications are required for the best outcomes after xenotransplantation, what types of genetic edits are required to prevent rejection, promote acceptance and good organ function in the human, how does the human immune system respond to pig organs, and does the pig kidney, for example, maintain normal function in humans, such as filtering urine and managing blood pressure. So in order to help bridge this gap and answer some of the critical questions, Dr. Montgomery and our team here at NYU have pioneered the pig-to-human decedent xenotransplantation studies, where brain-dead humans who are ineligible for organ donation and whose legal representative or next of kin authorizes thus to transfer the body and conduct these studies for a definitive amount of time. And I'll share a little bit about those experiences to date. So our team has conducted 5 pig-to-human decedent experiments, xenotransplant experiments at NYU. This includes 2 short-term gal knockout thymo kidney transplants that were maintained for 54 hours each. In these studies, we saw immediate graft function with no evidence of hyperacute rejection with minimal immunosuppression required that included corticosteroids and antimetabolites. We also followed up with 2 short-term 10 gene edit xenohearts in brain dead decedents that were maintained for 66 hours each. In both these hearts, we saw immediate graft function. However, in one of our experiments, we saw evidence of perioperative cardiac xenograft dysfunction. And that, after doing some more testing and research on what might have caused that, we saw that this could have potentially been mitigated by an incomplete T-cell depletion in one of our experiments where we only gave 1 dose of lymphocyte depletion as opposed to 2. And most recently, in July of 2023, we conducted a long-term xeno kidney where we transplanted a gal knockout thymo kidney in a brain dead decedent who was maintained for 61 days to assess organ function, immune response, and physiology. And in this experiment, we also saw immediate graft function and normal physiology for about the first month after transplant. After that, we found some evidence of antibody-mediated rejection, which we were actually able to successfully treat using FDA-approved medications that are found and used standard in clinical transplantation, which included a complement inhibitor, as well as plasmapheresis treatments, which is also standard of care for AMR in transplantation. It's important to note that none of the experiments here that we've done to date at NYU have utilized any additional investigational products aside from the genetically modified pig organ. Now I'll turn it over to Dr. Jayne Kim, who will discuss the xenotransplant living recipient cases to date.
Thank you so much, Dr. Khalil. So to date, there have been 4 patients who have been treated with investigational pig organs through expanded access. And this starts all the way back in 2022, which seems very, very recent. So in January 2022 marks the first pig to human heart transplantation at the University of Maryland. And then a year later, in September 2023, Maryland performed their second pig to human heart transplantation. And then 2024, this year, we actually had our first pig to human kidney transplantation. In March, Mass Gen, also associated with Harvard, performed their first kidney transplantation. And then shortly after, a month later, NYU Langone Health, our team here, performed our kidney transplantation as well. And so to give you additional insight on our specific experience revolving around NYU Langone Health in obtaining FDA approval for expanded access, we wanted to give you a little infographic to show you some quantitative facts that went to obtain FDA approval. So 2 things to note on this slide. Each case is different because each patient is different. And so this is just a portrayal of our experience at NYU Langone Health. We are not privy to Maryland or Mass Gen. And so this is our personal experience. And then number 2, something to note, the expanded access pathway that we use specifically is the single patient IND route. Dr. Kumar at FDA will be going over expanded access and what this is a little bit more in depth in his section. So I'll leave it at that. So first off, alongside the core transplant team, more than 50 individuals played a very crucial role in not only coordinating treatment, but also securing the FDA approval. So to provide insight into the breadth of interdisciplinary collaboration involved, key team members included professionals from the following areas. So we had infectious disease for obvious reasons, PT, nursing, we had psych, social work, palliative care, regulatory and IRB for regulatory approvals, surgeons, obviously, cardiologists, nephrologists. We also had the sponsor team who provided the organs to us involved as well. They had veterinarians, CMC staff provide information on the product itself. And then we also had labs across many different disciplines provide management of the tissues. So we had clinical and research involved there. And then we also had pharmacists, including Dr. Khalil, other pharmacists as well. And then, of course, scientists. While this is mainly for treatment, we also want to learn a lot from this expanded access treatment. And so we had scientists involved as well. Next, we wanted to give you an idea of how much documentation was actually needed for this specific FDA approval. So just to give you a little bit of information, typically a single patient IND is a fairly quick process. It could be from like 20 pages to 50 pages, nothing more than that typically. For our xenotransplantation case, however, it was greater than 2,500 pages. And so that not only highlights how complex this case was, but just the thoroughness of NYU Langone Health's team, as well as FDA's review team as well. And so to give you a little bit of more information on what type of documentation this included. So this does not include all of the forms that are required. This is actual patient information. And so patient history, we had letters from clinicians, and their opinions on the matter. We had an in-depth protocol and informed consent forms, and the protocol also included zoonotic monitoring processes. We also had a plethora of preclinical data, and something to note here, as Dr. Khalil introduced, we were able to use the decedent model data for approval for this case. And so that's something that we wanted to highlight and something that we found was very important here. And then not only did we have information about the patient itself, herself, we also had information from the preclinical data and the donor pig. And so the sponsor provided, in-depth veterinary diagnostic detail reports on the pig donor. Next, we wanted to highlight how long it took for us to obtain approval. So it took 30 days from initial submission to FDA approval, and that is within the review period that is defined by regulations. So typically INDs have 30 days for FDA to review and provide a decision, whether you are placed on clinical hold or approved. And so we just hit that deadline at 30 days. And then just to highlight how collaborative FDA was with us and how in-depth their review was, we had 8 total teleconferences with FDA within that 30 days. So they were very communicative with us, giving us guidance, and really pushing us and supporting us for this approval. Within those 30 days, we also had 9 information requests from FDA. So while we provided a pretty in-depth initial submission, there's always new information that FDA requested and wanted to review. This is based on, CMC data on the donor pig itself or the organ or patient history. So there were 9 of those. And then lastly, it took our team a total of over 150 hours spent on efforts to obtain FDA approval. So we just wanted to give you sort of a broad idea of our specific case in obtaining FDA approval for the xenotransplantation case.
I'm assuming that both of you are finished.
Yes, thank you, Dr. Caplan.
Okay, sorry.
Sorry, I should have introduced Dr. Kumar.
No, that's okay. So thank you for laying out that background and what has happened with respect to regulatory oversight and collaboration with FDA. Next, I've got Vijay Kumar. Dr. Kumar is with the CBER unit of FDA, and I'll let him say anything else he wants by way of introduction.
Good afternoon. Thank you, Drs. Caplan, Dr. Kim, and Dr. Khalil. My name is Vijay Kumar. I serve as a clinical team leader in CBER. It's my pleasure to share the FDA perspective on the use of expanded access pathway for xenotransplantation. Next slide, please. I have no conflict of interest to disclose, and my views do not bind or oblige the FDA. Next slide. The mission of the FDA is to advance the public health by helping to speed innovations that make medical products more effective and safer. The full mission is listed on our website. Next slide, please. To fulfill our mission, we are organized as multiple centers and offices across FDA with jurisdiction over review of different products. The review of the xenotransplant products fall under the jurisdiction of CBER, Center for Biologics, and Center for Veterinary Medicine. My office reviewing Xeno products, the Office of Therapeutic Products, is housed inside of CBER. Next slide, please. In OTP, we regulate a vast array of products ranging from gene and cell therapies, blood and plasma products, biologic devices, and of course, xenotransplantation. Yes, that's a long and a diverse list, and our super office has 6 sub-offices, 14 divisions, and 33 branches. Next slide, please. To fulfill our public health mission and shape our regulatory thinking, we continually interact and learn from the public, from academia and industry. Some of the areas of our learning are listed on this slide. Next slide, please. We regularly connect with the patient community because we want to be informed by the experience of those living with the clinical condition. In the Xeno space, we actively participated in the patient-focused drug development meeting organized by National Kidney Foundation in November of 2023. Next slide, please. At FDA, we regulate, but we do not develop drugs or biologics or conduct clinical studies. We continually interact with academia and industry through multiple channels. We value their feedback, recognizing that both of us share the same common objective of advancing bench research to bedside therapeutics. Next slide, please. Expanded access is a regulatory pathway. We know the term compassionate use is used colloquially in the mainstream media and also the topic of this webinar. Please also note that the terms drugs and biologics are used interchangeably in the regulations. Next slide, please. The aim of the expanded access pathway is to treat the patient. The primary purpose is not intended to collect data to support a marketing application. In the Xeno space, given the magnitude of unmet need for organ transplant as reviewed by Dr. Khalil and the complexity of the products, we believe the ideal approach to advancing this field is to conduct well-planned clinical studies under research INDs with a pre-specified and a structured approach for rigorous collection of clinical data. In this context, expanded access should be viewed as an initial first step or a bridge to advancing the field of xenotransplant. Next slide, please. We received 3 types of expanded access protocol based on the size of the population and the stage of the product development. As of now, the EA submissions allowed by the FDA fall under first type. Next slide, please. Some of the requirements for expanded access are similar to the research IND requirements, including protection of human subjects and IRB oversight for safety. Next slide, please. The focus of expanded access is on the patient. Therefore, we have 3 additional requirements: that the patient has a serious condition without treatment alternatives, the potential benefit justifies the potential risks, and the access to the product will not interfere with other investigations. As I will discuss in a subsequent slide, the term potential includes both known and unknown. Next slide, please. We have allowed expanded access treatments for using pig heart and pig kidney. Dr. Kim went over their perspective on the review from our side. Each of these submissions underwent intensive multidisciplinary reviews. We also engaged in extensive consultations with diverse subject matter experts housed throughout the agency as listed on the slide. These internal reviews were happening concurrently with comprehensive interactive discussions with the sponsor investigators and included both written and verbal feedback. During this process, various elements of the protocol and informed consent document were analyzed, revised, and refined multiple times. Next slide, please. Xenotransplantation has major public health implications with risk of zoonosis, not only for the patient, but also for close contacts and healthcare professionals from phlebotomists to surgical staff. Therefore, we require 3 separate informed consent documents or ICDs. We also require institutional participation with engagement of hospital epidemiology, infection control, and occupational health. Next slide, please. Yeah, this is a busy slide. On this slide, I would like to discuss some of the regulatory risk-benefit considerations that go into xenotransplantation. This list is not exhaustive and is not meant to represent OTP's review of any specific xenotransplantation product. Additionally, this list is primarily from a clinical perspective and does not contain all of the review considerations specific to other disciplines such as chemistry, manufacturing controls, pharmacology, toxicology, clinical pharmacology, bioinformatics, and veterinary medicine. Clinically, it is important that we define what is known and unknown about the potential benefits and potential risks of kidney xenotransplantation. There are currently a lot of unknowns regarding these risks. The potential benefit of the xenokidney transplant listed above are self-evident, including freedom from dialysis and freedom from vascular access interventions. We divide the potential risks into known, unknown, and unknowable. Known risks are those that are derived from published information in human transplant literature and available non-human primate xenograft data. We are cognizant that we do not have good models for chronic kidney disease. The NHP and human decedent data are based on a nephrectomy or acute kidney injury models. Although both NHP and human decedent models have complemented each other in filling the gaps in our understanding, there are certainly limitations in translating the observed risks to human recipients with chronic kidney disease. Some of these risks pertain to the growth of the kidney, species-specific physiologic mismatch, and durability of xenograft. Finally, without human studies, we cannot adequately characterize the unknowable risks of the zoonosis and the long-term impact of genetic modification. Therefore, we rely on surveillance during these first-in-human studies. At FDA, it is our stance that the informed consent documents clearly communicates both the potential risks and the potential benefits, while also discussing and acknowledging the current limitations of our understanding. As more safety and efficacy data become available from more patients followed for longer duration, our recommendations and guidance is bound to evolve over time. Next slide, please. This guidance document answers frequently asked questions on expanded access submissions. Next slide, please. This slide has links to the FDA guidance document on xenotransplantation and public health service guideline pertaining to risk of zoonosis. Next slide, please. Here we have links to our webinars, event, and different ways to connect with us. Next slide, please. In closing, we acknowledge there is a great deal of interest and excitement in developing the field of xeno organs. We are committed to advancing the field. We do not want to stymie the progress because of the complexity of the products and lots of unknowns, as discussed by me, Dr. Kim, and Dr. Khalil. Recognizing that the field is still nascent and evolving, we will take in any and all learnings from EA submissions to better inform us, even though we fully recognize that the intent of EA is to treat the patient and not for generation of data. Having said that, we know there are tens of thousands of patients with organ failure waiting for transplant. To reach scale, we encourage research INDs conducted with sufficient rigor to generate clinical data in supporting the continued product development towards an eventual marketing approval. I again want to emphasize that we do not control the type of submissions we receive from sponsors and investigators. We thank the CUPA NYU for providing us an opportunity to share the FDA perspective. We look forward to your questions. Thank you.
Vijay, just one clarification question that I see is come in. does the FDA, in looking at experts for consultation within the FDA, is there a consultation with outsiders, non-FDA in the US, or other regulatory bodies outside the US?
No, we have not kind of like, consulted for the purpose of the review experts outside the FDA or non-US bodies.
Thank you. All right, our final presenter, and I'm seeing plenty of questions. Remember, if you have a question, put it into the Q&A or the chat box. John and I will be watching both, so put your question in there and we'll get those queued up. My colleague Tamar Schiff has been working hard on issues around transplant and xenografting and expanded access, and I'm going to turn the floor over to her.
Thank you very much. Thank you to CUPA and to John Massarelli for organizing this, and Dr. Caplan for moderating, and of course my co-panelists. I kind of have the unfair advantage of having them set the scene and give all the necessary background, and I'm just going to make a few comments and raise a few questions, not necessarily answer them all. Just very briefly, I'm an internal medicine physician by training, and I'm a postdoctoral fellow under Dr. Caplan here at the Division of Medical Ethics. So as I mentioned, I really just want to focus on some ethical and other questions and issues raised by this intersection of xenotransplantation and the expanded access pathway. I'm sure we'll get into broader questions around clinical trials and such in the Q&A as well. So the first thing I just want to make clear is that I agree, as was put forth really wonderfully by all my co-panelists, that xenotransplantation should be aggressively pursued to try to mitigate the dire human organ shortage. And as was brought up, it could help not only those on transplant organ lists, but even those in earlier stages of organ failure, which is an immensely enticing prospect. The other thing I completely agree with is that FDA is absolutely the correct and trusted agency to be the ones regulating this. But I think the question at hand for today's webinar is whether the expanded access pathway is the right regulatory mechanism. And I think it's important to really sit with that, because it's not just a frivolous bureaucratic question. I think there are a lot of pros or advantages, as Dr. Kumar really wonderfully laid out for us, for using expanded access. It's an already available approach or mechanism, and as much paperwork and effort and time that Dr. Khalil and Dr. Kim mentioned it takes, it's not introducing, some new stack of paperwork that's required, because the mechanism is already in existence. But that being said, I think a huge gap between the expanded access pathway and clinical trials are the public reporting requirements. And I think in xenotransplantation, that's a really important thing to think about. Clinical trials have very extensive public reporting requirements through clinicaltrials.gov and other mechanisms. And that's really meant to ensure that all studies, positive and negative ones, are disseminated broadly to investigators, to clinicians, and then to patients, their families, and patient advocacy communities. And that's not the case with expanded access, which makes sense because historically or typically the pathway was meant to expand access outside of clinical trials. So this is certainly not every case, but typical case is a patient who's too sick to enroll in a clinical trial or otherwise ineligible, and then receives that still investigational medical product through expanded access for the purpose of treatment, not for the purpose of, as Dr. Kumar said, generating generalizable data for research. But here, I think this really kind of blurs the line, because this isn't really expanding access. This is first in human experience with something that is not under investigation in clinical trials. And that's not to say that's not the right approach, but I do think it's important to call attention to the differences from how expanded access has functioned to date, typically. So, as again, thank you to everybody who gave the background, and now I don't have to. We're many, many months out from the 4 cases of in living patient xenotransplantation that we've had here in the US, but only one of those cases has been reported in peer-reviewed literature, and it's really unclear when if at all, kind of the details, the clinical details, the pathology, potential adverse events will be available to the public, because that's not required under expanded access. And again, I'm not trying to say that that's categorically right or wrong. I just think it's a really important ethical, logistical, and even legal question that we need to consider. The FDA has this data, which again, I think is a really trustworthy entity, but other investigators, patients and their families don't have access to this data. So I think Dr. Kumar really laid out really wonderfully how this is kind of an evolution of the scope of purpose of expanded access, differently than it has been used previously. And if that is the case, or kind of this new direction, then I think the two really important things that are necessary are maximal transparency, as Dr. Caplan mentioned at the top of the webinar, and then a lot of thoughtfulness about the precedent this may set. So if expanded access is evolving into something that could sometimes be employed as kind of a pilot trial mechanism, a precursor, or as Dr. Kumar said, kind of a bridge to clinical trials, then we need to consider what that means, and also whether the reporting requirements need to evolve accordingly. And if we think about this kind of, again, being a new norm or a new precedent, how could institutions or corporations who are developing products, drugs, devices, how may they seek to use expanded access for first-in-human experience before clinical trials? I have no doubt that everyone involved to date, the investigators, the clinicians, the regulators, have been acting in very good faith, but I think it's important that a regulatory system doesn't depend on good actors, that they're really regulatory standards that ensure good action. And then the last point, I know I've just kind of raised a lot of questions, not answered them, so I hope that that really helps us get into the Q&A. But the last point I'll bring up is that we're still left with a lot of questions about starting clinical trials that this experience with expanded access doesn't necessarily help us answer. So the hesitations or some of the barriers to beginning clinical trials are around who should be first participants, what should be the eligibility criteria, how do we obtain true informed consent given the knowns, unknowns, and unknowables, as Dr. Kumar went through, how we can adequately monitor for and potentially contain zoonoses, and whether these organs should be bridges or destination therapies. And the expanded access experience doesn't necessarily elucidate any of those questions in a way that we could say, now we know who should be first participants in clinical trials. So again, I think that underscores that this really has been kind of a pilot trial mechanism, which perhaps is the right approach, but I just think if so, requires transparency about that fact and a lot of thoughtfulness and deliberate safeguards about how it could be used with future innovations for similar kind of pilot trial data collection. And then just as food for thought, maybe there's some middle ground or some evolution that could come out of this, where we have an expanded access like pathway with liberalized patient-specific eligibility criteria, but then strict reporting requirements such as clinical trials, so that this the data gathered, which is so crucial and is pilot data for many experiments to come, is disseminated more transparently. And with that, I'll hand it back to you, Dr. Caplan, for the Q&A.
Thank you so much, Tamar. These are really interesting presentations, I know, because I can see about 25 questions queued up there, ready to roll. So we will get into those, but I will ask a little bit of a clarification question again for Vijay. If you wanted to, as part of granting the IND and going through all this process, what is the authority of the FDA to either say you must then enter clinical trials, or you must then publish about what you do? In other words, post-approval of NYU work or Maryland or wherever it might be, what can the CBER and the agency do to impose further requirements after the fact, if any?
Okay. Thank you. As I kind of like, mentioned, at the FDA, we do not kind of like, control what submissions we receive, whether it is IND or expanded access submissions. Some of the sponsors on their own, including NYU, have kind of like, published their data in peer-reviewed kind of like, publications. To address to some of the issues that were brought up by Dr. Schiff, there is a very unique and unusual set of circumstances here that touch all three review disciplines. From a product side, it is obvious there's inherent complexity in implanting genetically edited organs followed by intensive immunosuppression. Science is still evolving on what are essential edits and what is optimal immunosuppression. From the preclinical models, although informative, are imperfect, and the translatability of the findings to human study is not clear. From the clinical side, there is tremendous public health interest, not only in addressing this organ shortage, but also public health concerns with the risk of zoonosis. So the EA pathway has the potential to address our knowledge gaps in all three review disciplines. However, again, I would like to state that to advance this field further and reach scale, there is a need for rigorous collection of data under a research IND.
And I can't resist a follow-up there. Collecting it and publishing it, making it available, posting it. What's the transparency side?
On the, it is a very good question, and we wrestle with that inside the FDA. There are obviously public health interest and public health considerations. We are a public health agency. At the same time, we are also bound by confidentiality with the sponsor investigators, and particularly in the expanded access case, since the focus is on the patients, there are also additional HIPAA regulations. So considering all of this stuff, yes, we try to kind of like, make information available as much as we can under the existing rules and regulations.
I've got one question here, Jayne or Karen, I'm not sure who wants to take it. It's kind of a interesting short one, but in trying to work with the FDA and seeking approval, what do you think was the toughest hurdle you had to surmount? And where were they surprising in any way in the pushback that they gave?
I can start this off, and then Karen, if you want to jump in. From my perspective, I think it was all of the preclinical data that was required from our team. So obviously non-human primate data was just not available using the specific kidney for our case. And so, just finding sufficient information to relay the safety profile of the organ and mitigate risks that way, I think was the biggest hurdle. Karen, I don't know if you have any other input on that.
Yeah, I certainly agree. And I would say, I think our case is unique in where we were able to actually submit all the raw data from our decedent study, and that was reviewed, and that was what was considered in the decision to approve our single patient IND, which is, I think, unique to our scenario compared to the other living humans that were accepted.
And I've got a follow-up. You may or may not know the answer to this, but I'll try it on either of you. Some kidney teams have suggested, as have some people interested in xenograft, that when you give consent, are you getting consent from the research participant to come off the human list? Is it only people who would be ineligible to get a human kidney who could receive it? And if so, why?
So in our scenario, and maybe in the previous experiences, these were patients who were either ineligible for human organ transplantation, or their risk of mortality on dialysis outweighed the potential risks and unknown risks associated with the xenotransplant experiment. And I don't think that just the people who are ineligible for organ donation moving forward should be the ones we consider. I really think in thinking about equipoise, you really have to see who would benefit the most from these organs. And, you know, as we continue to learn more, it's important to really evaluate our wait list and assess what the optimal candidate here would be, and not just those ineligible for organ transplant.
Thank you. Why, and this is to anybody who cares to comment, but there's just a simple question. Why are we doing kidneys first? Is that because the shortage is biggest, or is it biology?
I would say it's definitely the organ that's most in demand right now. But I think, you know, there is now experience with kidneys and hearts, and I know there will be future preclinical studies looking at other types of organs and maybe other types of therapies that may bridge patients waiting transplant.
And here's what I think more for Vijay and Tamar. So they congratulate NYU for gaining EA approval. I think they're saying against a very tough uphill partner at the FDA, but they didn't say that. I said that. But what are the plans, if any, for sharing templates and documents so that other academic groups who want to develop their processes can do so? This gets to the transparency issue that Tamar raised. There certainly are other groups, both in the US and elsewhere. I should mention, by the way, I'm seeing questions from around the world here. So people are watching in other nations. So are we going to have to go back and do it, you know, reinvent the wheel every time?
So I think it's really difficult to create a standard template for these type of cases, especially in the single patient IND world, because every single patient diagnosis or their conditions differ. And so what FDA is going to want to hear about the patient is going to differ every single time. So information requests are going to differ. FDA might ask for additional clinical information requests or additional CMC information requests. And so to create that golden standard template at this time, I do foresee it being challenging. However, you know, there's always that standard single patient IND template that we always start off with. So yeah, I think at this time, a bit challenging to provide a finalized template for everyone to share.
Yeah, thank you. Thank you, Dr. Kim. From the FDA perspective, as I indicated, we are a learning organization, and we are starting off of complex products with imperfect animal models. And for example, till the rubber hits the road, we don't know whether the anastomosis will hold up against the hypertension in a patient with chronic kidney disease. And our understanding and thinking continues to evolve, and the science is still not yet settled for us to come up with any kind of a formal kind of template. And based on our learnings, the entire FDA team may be willing to kind of relax some requirement based on sufficient knowledge of safety, or maybe even willing to kind of request additional information when we kind of see certain safety signals. I also want to point out that the sponsors have been extremely collaborative with us, not only up to the point of the review, but even after the transplants, to share the continued clinical course of the patients, so that we are informed much better. And I really want to thank all the sponsors and investigators for continuing to collaborate with us. Thank you.
And if I can add just one thing, I will say in my experience over the past 3 years here as a xenotransplant project manager, we've actually collaborated with numerous institutions across the country and have signed agreements, non-disclosure agreements, and shared a lot of the work that we've done to help other centers set up similar types of studies in the decedent model to help them gain the preclinical information that we're able to conduct here. And our goal here is to get as many people transplanted as possible and to really move this field forward. So, we're not trying to practice here in a silo. And I think from the IND perspective, our team's goal is to publish, comprehensive, high quality literature in peer-reviewed journals as soon as possible, but that does take a lot of time. But in the meantime, we're always open to sharing, our experiences and what we've gone through to help others do the same.
I think a question that is coming up here that's very interesting is, to what extent are industry sponsors involved in this type of work? I'll call it. I don't want to call it research or access, just initial going into humans in the context of, investigational procedures. We do have some reason to worry that some industry sponsors might play expanded access a little bit to try and get earlier approval or to hype something. And in the background here, we have not only issues around who's sponsoring the xenograft, whether it's NYU or university, but also these genetic engineering companies way in the background that are working with the pigs. So how is that managed or thought through in terms of conflict of interest, or making sure that, there isn't too much hype?
No, I think I can answer from a scientific and a regulatory perspective, but this question is really out of our wheelhouse.
I would say in our experiences, the IND holder, or in our experience, the IND holder is the investigator. The sponsor supplies us with the pig organ, but in terms of a conflict of interest, the IND holder and the person conducting this type of procedure is actually the investigator and not the sponsor.
And that leads to another question. What role, if any, did the IRB at NYU play? Did you compute their hours in your little chart? And should IRBs play a role here, or are we in a gray zone?
Yes, so Kieran and I can take that one as well. So obviously IRB plays a huge role in this, right? Because they have to approve the this case itself as well. And so there was a lot of coordination between regulatory, the transplant team, and IRB. So shout out to Marina, who is the director of our IRB. And so a lot of hours of communication, and yes, that was factored in the total amount of hours here. Kieran, if you wanted to add anything about IRB's involvement.
Yeah, the IRB is integral to this. I mean, they, very thoroughly review the very hefty packet that we submitted to the FDA to ensure that we're doing everything according to appropriate ethical standards and regulatory standards. And I would say actually, even for our decedent studies, we actually have an IRB equivalent committee called the Research on Decedent Oversight Committee that approves all of those protocols, the same type of process that you would undergo for any IRB study. So all of these are considered and approved with the same type of rigor.
And why is that? Why a special committee?
So when we were first initiating the idea of doing these studies, the fact that these decedent subjects are deceased and they're not technically, by that definition, human subjects, it didn't fall under the purview of the IRB. And therefore, our group created an IRB equivalent that just looks at and reviews any research that comes through that's proposed in the brain dead decedent model called the Research on Decedent Oversight Committee.
And here comes one for Tamar. Why not just say, let's go to clinical trials from the get-go?
That is the question I wanted to raise. I think to bring it back for a second to the involvement of the sponsors, I do really appreciate your answer, Dr. Khalil, that the IND holder is the investigator. But it does open the door to sponsors who have relationships with investigators using this pathway, and again, accruing pilot data for their downstream clinical trials without the burdens of those initial clinical trials. And again, I think everyone has been a good faith actor, but that potential precedent, I think, is one we need to be very thoughtful and deliberate about and create safeguards for. So I do want to mention that important consideration with the sponsor involvement. And then, I messaged Dr. Caplan separately that I do want to raise this point, I'll give my thoughts, but I would be really interested in the rest of the panelists' thoughts. Why, if are using this as a bridge, the expanded access pathway, if it is giving us valuable data, though it's not being shared like clinical trial data for investigation, for formal investigation down the line, why not start a formal, if it's a single patient or multi-site few patient clinical trial, and then it'll be subject to clinical trial reporting requirements and the same sort of safeguards that make clinical trials trustworthy and guarantee safe and effective innovations.
And kind of in the same vein, what sort of requirements are appropriate for follow-up with this patient? Does the FDA have any ideas about what it would say to anybody who's seeking this pathway right now in terms of monitoring for zoonoses or whatever, lab testing down the road and so on?
Yeah, thank you very much. As a part of the protocol, there is a schedule of assessments which kind of outline assessments along multiple domains, including zoonosis surveillance and different reporting periods for all of those surveillances. They're all laid out in the sponsors and in the investigators protocol. In response to Dr. Schiff, yes, I completely agree that this is not an ultra rare condition. This is the end organ failure of tens and thousands of the patients. And to really advance the field and move the needle on xenotransplantation, we need research INDs. Having said that, we do not control the submissions that we receive from sponsor investigators, and we review each submission rigorously from a scientific and a regulatory standpoint. But your points about transparency are very important, and we do take it seriously. Thank you.
And one thing I just want to add is that our goal is to move to clinical trials. Our goal is not to continue doing these as one-off single patient INDs. And I think, in working with the FDA and understanding what that requirement will look like to submit that, pre-IND and what the data that they're willing to accept is to allow us to move forward to clinical trials, I think will be a really important collaborative effort.
So let me do this. Plenty more questions. Apologies if I didn't get to yours. If you wish to send us the question offline through an email to me or to John, we can try to get them answered by our speakers or find someone who can answer them. But I do want to give each of the presenters a last 45-second opportunity to say anything they might wish to add or something that they didn't get asked that they want to make sure they make a point about. You don't have to do that, but if you want it, Jayne, you have the floor first.
Thank you, Dr. Caplan. Yeah, so just to kind of wrap everything up, we just wanted to sort of impress upon the participants here that we are all in this journey together with the same goal of helping these patients who need this, who are in dire need of this xenotransplantation. And so, ethics, FDA, IRB, regulatory affairs, transplant team, we're all in this together. And I just wanted to highlight that. And thank you so much for having us again.
Yeah, thank you for the opportunity to share the FDA perspective. And I have to stress that it's, we have collaborated in the spirit of advancing the field of xenotransplantation. The sponsors have been open, frank, interactive, and have significantly helped us shape our thinking on review of each and every subsequent product. Thank you.
Of course, that's related to the presence of the bioethics unit as a partner in all of this. Only kidding. But we have at NYU a good environment from the time at which we first proposed research on the newly dead as a way to gain data all the way out to thinking about working with the IRBs and special committees and so on. So it's been a good relationship here in terms of environment. Tamar, you get the last word.
Okay, I just want to second what was said by everybody else, especially Dr. Kim, about the really true collaborative effort. And in that spirit, just say about whether there should be, raise the question of whether there should be, similar kind of hive mind approach to a more appropriate or evolved regulatory approach to these types of innovations. I think xenotransplantation just raises that this will perhaps come up more often as we continue to innovate outside of xenotransplantation as well, and whether there should be some hybrid or new mechanism that doesn't heighten the bureaucratic load, but does allow us to innovate speedily while still maintaining the trustworthiness and the transparency that the field requires.
And I said you had the last word tomorrow, but I'll end on this note. With a new administration coming in, and we're very familiar, we are at NYU, with a lot of discussions about ways to gain speed in innovation, it may be important in that context to think about what would be the best approach, if not the current approaches to IND and making progress that way rapidly, moving to clinical trials, or another path, which might, again, reinforce what I think will be a message that comes through pretty clearly that, innovation and speed ought to be encouraged. I'm thinking from interaction with the previous Trump administration that was pretty clear. So that's the context, at least in the US, that we're likely to see come forward. So think about that in that context. Again, welcome any emails, actually welcome any feedback you want to give us. You thought this was useful and important. I thought the presentations were really illuminating. It's great to get a peek behind the scenes at what's going on and how the thinking goes. That sort of transparency is wonderful. And I appreciate the audience coming and sharing this time with us. And thanks again to John Mazzarelli for putting this together. So thanks, everybody. Have a good day.
Thank you.